Dear viewers, this video is part 1 of the 7-part series on lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. WBCs, lymphocytes and immunity. The human body has the ability to resist almost all types of organisms or toxins that tend to damage the tissues and organs. This capability is called immunity. Much of the immunity is acquired immunity that does not develop until after the body is first attacked by a bacterium, virus, or toxin. Often, weeks or months are required for the immunity to develop. Innate immunity. Innate immune system, ancient immune recognition system of host cells bearing germline encoded pattern recognition receptors PRRs that recognize pathogens and trigger a variety of mechanisms of pathogen elimination. Cells of the innate immune system include natural killer cell NK cell, lymphocytes, monocytes bar macrophages, dendritic cells DCs, neutrophils, basophils, eosinophils, tissue mast cells, and epithelial cells. Note: germline encoded means already existing recognition pattern built into the genetic code during development of the fetus. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 1 lakh prescriptions. Innate immunity includes the following aspects: phagocytosis of bacteria and other invaders by white blood cells and cells of the tissue macrophage system, as discussed in granulocytes and granulocyte functional aspects in Medicine for Anyone in YouTube channel videos. Pattern recognition receptors PRR, germline encoded receptors expressed by cells of the innate immune system that recognize PAMPs. Pathogen associated molecular patterns PAMPs, invariant molecular structures expressed by large groups of microorganisms that are recognized by host cellular PRRs in the mediation of innate immunity. Destruction of swallowed organisms by the acid secretions of the stomach and the digestive enzymes. Resistance of the skin to invasion by organisms. Presence in the blood of certain chemicals and cells that attach to foreign organisms or toxins and destroy them. Some of these are: 1. Lysozyme, a mucolytic polysaccharide that attacks bacteria and causes them to dissolute. 2. Basic polypeptides, which react with and inactivate certain types of gram-positive bacteria. 3. The complement complex, described later, a system of about 20 proteins that can be activated in various ways to destroy bacteria. And 4. Natural killer lymphocytes, NK cells, that can recognize and destroy foreign cells, tumor cells, and even some infected cells. Innate lymphoid cells, ILCs, lymphocytes that do not express the type of diversified antigen receptors on T cell and B cells. ILCs 1, 2, and 3s are tissue resident cells and functionally are analogous to CD4 Th1, Th2, and Th17 cells respectively. Natural killer cells, NK cells, a type of ILCs, that is innate lymphoid cells, that kill target cells expressing few or no human leukocyte antigen HLA class I molecules such as malignantly transformed cells and virally infected cells. Innate NK cells mirror the functions of CD8 cytotoxic T cells of the adaptive immune system. Natural killer or NK cells are a small but important blood lymphocyte population constituting only 2%, distinct from T cells and B cells. They do not express the T cell receptor for antigen or CD3. They do not express the surface immunoglobulin B cell receptor. In contrast to adaptive immune responses mediated by T cells, these ILCs have the innate natural killer cells have the innate ability to kill target cells without prior sensitization. This is known as natural killing. They therefore play a key early defense role against many infectious pathogens. Dendritic cells, DCs, myeloid and or lymphoid lineage antigen presenting cells, APCs of the adaptive immune system. Immature dendritic cells, DCs, or DC precursors are key components of the innate immune system by responding to infections with production of high levels of cytokines. DCs are key initiators of both of innate immune response via cytokine production and of adaptive immune response via presentation of antigen to lymphocytes, T lymphocytes. Immunity at mucosal surfaces. Mucosa covering the respiratory, digestive, and urogenital tracts, the eye conjunctiva, the inner ear, and the ducts of all exocrine glands, containing cells of the innate and adaptive mucosal immune system that protect these surfaces against pathogens. In the healthy adult, mucosa-associated lymphoid tissue, MALT, contains 80% of all immune cells within the body and constitute the largest mammalian lymphoid organ system. Note: exocrine glands, a gland that makes substances such as sweat, tears, saliva, milk, and digestive juices and releases them through a duct or opening to a body surface. Examples of exocrine glands include sweat glands, lacrimal glands, that is tears, salivary glands, mammary glands, that is breast milk, and digestive glands in the stomach, pancreas, and intestines. MALT has three main functions, that is mucosal associated lymphoid tissue. One, to protect the mucous membranes from invasive pathogens. Two, prevent uptake of foreign antigens from food, commensal organisms, and airborne pathogens and particulate matter. And three, to prevent pathologic immune responses from foreign antigens if they do cross the mucosal barriers of the body. MALT is a compartmentalized system of immune cells that functions independently from systemic immune organs. Whereas the systemic immune organs are essentially sterile under normal conditions and respond vigorously to pathogens, MALT immune cells are continuously bathed in foreign proteins and commensal bacteria. They must select those pathogenic antigens that must be eliminated. MALT contains anatomically defined foci of immune cells in the intestine, tonsil, appendix, and peribronchial areas that are inductive sites for mucosal immune responses. From these sites, immune T and B cells migrate to effector sites in mucosal parenchyma and exocrine glands where mucosal immune cells eliminate pathogen-infected cells. In addition to mucosal immune responses, all mucosal sites have strong mechanical and chemical barriers and cleansing functions to repel pathogens. Key components of MALT include specialized epithelial cells called membrane or M cells that take up antigens and deliver them to dendritic cells, DCs, or other antigen presenting cells, APCs. Effector cells in MALT include B cells producing anti-pathogen neutralizing antibodies of secretory immunoglobulin A as well as IgG, immunoglobulin G. See later. T cells producing similar cytokines as in systemic immune system response, and T helper and cytotoxic T cells that respond to pathogen infected cells. Secretory IgA is produced in amounts of more than 50 mg per kilogram body weight per 24 hours and functions to inhibit bacterial adhesion, inhibit macromolecule absorption in the gut, neutralize viruses, and enhance antigen elimination in tissue through binding to IgA. Recent studies have demonstrated the importance of commensal gut and other mucosal bacteria to the health of the human immune system. Normal commensal flora induces anti-inflammatory events in the gut and protects epithelial cells from pathogens through toll-like receptors, TLRs, and other pattern recognition receptors, PRR signaling. When the gut is depleted of normal commensal flora, the immune system becomes abnormal with loss of Th1 T cell function. Restoration of the normal gut flora can re-establish the balance in T helper cell ratios characteristic of the normal immune system. Diet also has an impact on the gut microbiome. Altered microbiome composition has been etiologically related to obesity, insulin resistance, and diabetes. When the gut barrier is intact, either antigens do not transverse the gut epithelium, or when pathogens are present, a self-limited protective malt immune response eliminates the pathogen. However, when the gut barrier breaks down, immune responses to commensal flora antigens can cause inflammatory bowel diseases such as Crohn's disease and perhaps ulcerative colitis. Note: Crohn's disease is a type of inflammatory bowel disease, IBD. It causes swelling of the tissues, inflammation in the digestive tract, which can lead to abdominal pain, severe diarrhea, fatigue, weight loss, and malnutrition. Ulcerative colitis is an inflammatory bowel disease that causes inflammation and ulcers, sores in the digestive tract. Ulcerative colitis affects the innermost lining of the large intestine, that is colon and rectum. Uncontrolled malt immune responses to food antigens such as gluten can cause celiac disease. Note: celiac disease is an immune reaction to eating gluten, a protein found in wheat, barley, and rye. Over time, the immune reaction to eating gluten creates inflammation that damages the small intestine's lining, leading to medical complications. It also prevents absorption of some nutrients, that is malabsorption. The classic symptom is diarrhea. Other symptoms include bloating, wind, fatigue, low blood count, that is anemia, and osteoporosis. Many people have no symptoms. The mainstay of treatment is a strict gluten-free diet. Acquired, that is adaptive immunity. In addition to its generalized innate immunity, the human body has the ability to develop extremely powerful specific immunity against individual invading agents such as lethal bacteria, viruses, toxins, and even foreign tissues from other animals. This ability is called acquired or adaptive immunity. Acquired immunity is caused by a special immune system that forms antibodies and or activated lymphocytes that attack and destroy the specific invading organism or toxin. Acquired immunity can often bestow an extreme degree of protection. For example, certain toxins such as the paralytic botulinum toxin or the tetanizing toxin of tetanus can be protected against in doses as high as 100,000 times the amount that would be lethal without immunity. It is for this reason that the treatment process known as immunization is so important in protecting people against disease and against toxins, as explained later. Dear viewers, this is the end of part 1 of the 7-part series on WBCs, lymphocytes and immunity in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.